Good afternoon, and for those of you joining us from across the country and around the world, good morning and good evening. Welcome to our webinar on the lung microbiome. I'm Amy Lietman, President of NTM Info and Research, and I will serve as moderator for today's session of Science from Your Sofa. On behalf of all of us at NTMIR, thank you for joining us. Our topic today is the lung microbiome, and we are delighted to welcome Dr. Leopoldo Segal and Dr. Benjamin Wu as today's featured guest speakers. Dr. Segal is the director of the Lung Microbiome Program and an associate professor of medicine at NYU Grossman School of Medicine. He received his MD from Universidad Nacional de La Plata, Argentina, and his Master's of Science from New York University. He is licensed in pulmonary medicine and critical care medicine. Dr. Segal is the principal investigator of multiple projects obtaining lower airway samples through research bronchoscopies, as well as through clinically indicated bronchoscopy. These projects include subjects with different pulmonary conditions wherein multiple culture-independent assays can be used to uncover bacteria presence in the lower airways and elucidate how they affect the host immune tone. Dr. Benjamin Wu is an instructor of medicine in the Division of Pulmonary, Critical Care, and Sleep Medicine at NYU Grossman School of Medicine. His interest in research started in the laboratory of Dr. Melissa Spencer at the University of California, Los Angeles, where he was trained in laboratory science techniques and inspired to pursue a career in translational research to dissect mechanisms. During his residency training, he pursued pulmonary research with Drs. Leopoldo Segal and Martin Blazer. His research focuses on the use of in vivo murine models and ex vivo microbiological methods to assess host microbial interactions. Dr. Wu has expertise in the use of bioinformatic tools to dissect the model organisms and their response to human oral commensals, and continues to work in human clinical trials. During today's webinar, you may ask questions using the Q&A function on Zoom. Click on the Q&A icon on your screen, type your question into the box, and then click send. At the conclusion of the presentations, the moderator will read questions from the queue for Drs. Segal and Wu to answer. Today's webinar broadcast is being recorded and will be posted online at a later date, so if you miss the answer to a question that is asked, you will have the opportunity to hear it again. Our next webinar topic will be hollow fiber research on Tuesday, April 20th, with guest speakers Dr. Jaco van Ingen and Jelmer Rijmakers from Radboud University Medical Center in the Netherlands. Registration will open next week. You can learn more about NTM Info and Research and sign up for our electronic news at ntminfo.org. And you can also find us on Instagram, Facebook, Twitter, YouTube, and LinkedIn. And now I'd like to turn this event over to Dr. Leopoldo Segal as our first speaker, who will later be joined by Dr. Wu. Dr. Segal and Dr. Wu, welcome. Thank you for both for making the time for our educational series. And although you are in the process of an important study and have been taking a lot of important meetings about it, thank you for taking time out of your very busy schedules today to join us.
Thank you very much for the invitation. It's a pleasure to be here. So my lab focuses on microbiome in many different conditions, and NTM has been one that we've been paying quite a bit of attention. But we do this taking the opportunity that we have at NYU, where we have started a multidisciplinary program that we call it the bronchiectasis multidisciplinary program, that involves not just pulmonary, but also other specialties such as GI, rehab, radiology, microbiology, really fostered by three clinicians and clinical investigators, Dr. Adris Harris, Kamal Masavaraj, that are really have helped tremendously to put together a very large cohort of patients. Yeah, and in that cohort, we focus on excellence of patient care, but also fostering research, a lot related to therapies and clinical trials, and a lot related to mechanism, understanding the mechanism of this disease. We don't do it on isolation. We do it in collaboration with the COPD Foundation. And this is a consortium of many academic places across the country that are represented here, we're one of the largest in terms of patient cohort and longitudinal follow-up that we have. So what we're gonna try to um, introduce today and try to give you a better understanding relates to this type of research that we do on molecular mechanisms, trying to understand pathophysiological things that occur in patients with bronchiectasis and NTM. So for that, the focus is on microbes, and I'm gonna let now Dr. Wu take over to give you the introduction.
Thank you, Dr. Segal, and it's a pleasure for me to be here today to speak to all of you at the NTM Info and Research groups. And I am very excited to start the discussion on the lung microbiome. So, well, my name is Benjamin Wu. I am an assistant professor of medicine, and thank you again so much for the time to discuss some of the core concepts behind the lung microbiome. I want to start out by addressing some of the more general aspects of disease. So traditionally, and maybe historically even, the idea of disease has been one pathogen causing disease. Things like pneumonia is caused by Streptococcus, E. coli causing urinary tract infections. But not all bacteria are necessarily bad. We have a large amount of bacteria in our mouths and in our gut that we call commensals. And these commensals, by definition, share our body with us. They are in our mouths, and they don't necessarily benefit us as far as we know historically, but we give them a place to live, to sort of colonize. And generally, a healthy commensal microbial community is associated with health. However, our understanding of disease has been upended with the discovery and the description of the microbiome. So now that we know that things like the pathogens do not exist solely as a isolated bacteria. In fact, oftentimes pathogens can be associated with a community of organisms and associated with commensals or oral commensal like bacteria. And when this community gets disrupted, it can lead to disease. And so our understanding of this sort of the cause of pathogens leading to disease has been upended by our understanding of the microbiome. So what is the microbiome? Broadly defined, the microbiome is a collection of microbes, including viruses, bacterial, and fungi that live on or in the human body and includes their activities. And so let's start the story with our previous understanding of the lung microbiome. In front of you is a picture of Robbins' textbook. And if you ask any medical student of our generation, Dr. Segal's generation, we probably own 1 or 2 copies of this textbook. It's about 1,500 pages long and describes how disease happens in the body. And what's unique about it, it has been that basically, up until around 2010, the normal lung has been free from bacteria. And this was an assumption that was made because any bacteria or any microbe isolated from the lungs was considered to be a pathogen. And now we know that's not necessarily true. And so from the discovery of the microbiome, there's been a virtual cornucopia of publications about how to unlock its potential, how the microbiome affects your health. For example, this article here about vaping can change your oral microbiome, leading to deleterious health effects, to modulation of your gut microbiome to help you age. And so you can see that everybody's been very, very interested in how the microbiome leads to health and disease. And in particular, our understanding, again, has been very, very black and white. pathogens lead to disease based upon how the body affects the immune tone. And so this crosstalk between your body and a potential bacteria being a pathogen leading to pneumonia can either be, escape, where this pathogen can lead to disease, or your body can successfully fight off the pathogen. And what we've discovered is that the microbiome, especially in the lung, can crosstalk with the immune tone, and the immune tone in your body can talk to the microbiome. And in turn, that can lead to how the body or even the microbiome interacts with the pathogen. With a healthy microbiome, it may suppress the ability of the pathogen to cause disease. However, with a dysbiotic microbiome, a microbiome in disarray, that may lead to increase in disease or the ability for the pathogen to cause things like pneumonia. And so where does this microbiome from the lung come from? And so we have two locations that are of interest: that of the upper airway, the oral and nasopharynx, and that of the gut. And what we've discovered so far is that the oral and upper airway is in direct communication with the lungs. It's one long tube from the mouth all the way into the lungs. And we think that the movement of these commensals, or the bacteria that reside in your mouth, go into the lungs. And this is seen over multiple studies, even in healthy people, that there is this idea that there's a microaspiration, or this communication between the oral and the lungs, and this movement of bacteria into the lower airways. However, not enough to cause disease. The other aspect is that of the gut-lung axis, and the gut microbes can also influence lung health in sort of a secondary way. And we call this immune gateway, because we think that instead of direct translocation of bacteria from the gut into the lungs, the gut microbiota can influence lung health through signaling molecules and inflammation. And we call this the gut-lung axis. Because there is a direct communication between the nasal and oral pharynx, there may be a microbiome gateway where what we normally do, such as breathe or talk or speak, we can shower the lower airways with our oral commensals. And this may lead to differences in how the body or the lungs react to health and disease. And so we also call this the lung-lung axis. And so there are many other factors that can influence your microbiome. And you can see this is a schematic that was published in Nature Review Microbiology from Dr. Bogart's lab by Men et al. And you can see that it can start at your time of birth and your genetics, the genes that you get from your mother and father, to how you were born, to who your siblings were, if you went to daycare, if you were exposed to antibiotics when you were younger, and if you were smoking or exposed to secondhand smoke growing up. And that may lead to perhaps a balanced microbiome that is stable and resistant from infection and resilient against other pathogens. Or maybe there could be a dysbiotic or an altered microbiome that may be in imbalance and less stable and more susceptible to infections. And so this is sort of the overall, or attempts at understanding how the lung microbiome can influence disease. So who are we, and who are this microbiome, this consortium of bacteria we have in our body? Overall, there's about 10 to the 13th human eukaryotic cells, but it's estimated that in fact there's probably 10 to the 14th bacterial cells, about a 10 times more increase than our human eukaryotic cells. And you have to wonder, if we are in fact more bacteria than we are human, how do we define who we really are? And not only is the fact that we have lots of more bacteria in our body than human cells of importance, the bacteria have many, many more genes that we are just beginning to understand. It's an estimated that compared to the 23,000 human genes that are known, bacteria contain about 8 million bacterial genes, of which only a small fraction of which we understand their function and their role in things like metabolism and disease and health. And so that's about 300 times more bacterial genes than human genes. So again, this large well of genetic potential. And so how do we define the microbiome? How do we identify what is in our lungs and in our organs? And so traditionally, what you have here, again, going back to our pathogen, we often will get a sample from, let's say, sputum or the gut or a stool sample, and that particular pathogen that's defined by these purple cells on the screen may come in the sample that contain many other bugs, many other microbes. And we traditionally depend on microbe isolation using things like culture, like growing them on a petri dish, growing them on liquid media. However, this culture-dependent method is very precise. And in fact, fastidious organisms are very hard to grow without the right mixture, without the right chemicals, without the right substrates, are actually very difficult to isolate using these methods. So very recently, what we've discovered is that we can actually use bacterial DNA or bacterial products or bacterial RNA to define this community of bacteria that may be in sputum, it may be in stool, it may come from a sample of the lower airway and lungs. And we call these methods culture-independent methods. Now, specifically, there are many different ways that these microbial byproducts and molecules can inform the community. And so amongst which we can isolate microbial DNA from samples that can tell us who or what species there are, who is not there, and what genes may exist. So we can tell what genes that exist in the lower airways. And in terms of the science behind that, we call that microbiome. So basically identifying the community members of a sample. And then if we really want to get technical with it, we can understand the kind of genes they have through the metagenome. So the metagenome is not only identifying the species or the identity of the bacteria in this collection or the sample, we can actually identify the genes that exist there. However, we cannot tell the function. We don't know if they're being expressed. We can, on the other hand, isolate even microbial RNA. And as you know, RNA is the next step. It's translated DNA. It's about to become a protein. And isolating the microbial RNA allows us to see what is being expressed. So previously, we can only identify the genes, but isolating the RNA, we can actually see what's being expressed by the bacteria. And we can also tell if they have active metabolism, so they're alive and producing this RNA. And the omics version of this is called the transcriptome, taking the transcripts and being able to isolate them. We can also identify and collect proteins, and that's called the proteome. That's not necessarily a specialty of mine, but this is certain labs can also identify proteins. And we can also look at metabolites, and these are the end products of the bacteria. So bacteria that can produce certain things like short chain fatty acids or cell wall components such as lipopolysaccharides. And this is sort of the cutting edge of what we can do nowadays, where we can isolate these small molecules that bacteria produce. And that we call the metabolome. And so overall, I want to tell you, this is a figure that was we took from one of the first publications in 2012 of the microbiome. And you can see two interesting figures. The left-hand figure is basically what they call a plot, a PCOA plot, a principal component plot. And each dot on this plot represents a sample color-coded by the location it was obtained in. So you can see that the blue are gastrointestinal samples, the oral are orange, and these are oral samples. But you can tell that every location has a unique microbial signature, and they cluster very uniquely apart. And you can make the argument that maybe the nasal overlaps with the skin, but that's what we understand about it. On the right-hand side is a schematic of a person, and they break down the different microbiomes across their body locations. And what you have to notice was in 2012 that they did not identify the lungs as a potential site that would harbor a microbiome. And that has since been disproved. And we now know that there is a lung microbiome. And so what do we think may happen in health and disease? And in front of you is a schematic. And what I want to go over is these broad concepts of what health and disease is in relation to what we understand about the lung microbiome. And the curved area at the bottom is essentially the alveolus of the lung, and this is the conduction airways, and below is a capillary that runs along the alveolus. During health, we know that when we microaspirate or we bring bacteria down from our oral pharynx down into the lungs, that these bacteria are very transient. They actually are cleared because we have a lot of protection from our immune system. Cells such as macrophages clean up this bacteria, but we also have something called the mucociliary ladder that basically are these hairs on our cells that beat up and bring out debris such as pollution, but also bacteria out of the airways. And so all these, the lung's ability to clear the bacteria are relatively intact, and there's not a lot of inflammation. Everything works as intended. However, when you have issues in the lower airway, say things like bronchiectasis or a disrupted airway, there is disruption of this mucociliary clearance. And there may be a buildup of these oral commensals in the lower airway, resulting in things like increased inflammation, macrophages that are pro-inflammatory, meaning they are angry and stimulate the immune system and stimulate inflammation. And to bring in things like interleukin-8, which is the arrow here, and it allows things like neutrophils, which are these very, very inflammatory cells, these white blood cells, to try to clear out this infection, to clear out the oral commensals that build up in the lower airway. And this is how we view disease in context of a disrupted or dysbiotic lung microbiome. And so what we've started to try to do from my translational experiments is try to understand what happens in the context of aspiration. And so this is a publication that was recently accepted. And in it, what we tried to do was model aspiration to understand what happens to the lung microbiome and the lung immunity. And what we found was that when we make mice aspirate, and mice don't aspirate, but we use a model where the mice aspirate human oral bugs, human microbes, to change their lung microbiome. What we saw was that they quickly recruited, as we'd expect, neutrophils and macrophages and T cells to come clear out the bacteria. And in some respects, a single aspiration event actually may be protective against certain infections, such as strep pneumo in this case. However, what we're trying to understand is that no one aspirates just once. And in many people with chronic aspiration, where people keep aspirating, we hypothesize that they will have an altered lung microbiome and altered lung immunity that make them more susceptible to lower airway infections and pathogen susceptibility. And that's currently one of the questions we're investigating in Dr. Segal's lab. And one of the things we're really trying to understand about how the lung microbiome may help or hurt you in terms of pathogen and infection. And so I want to leave you with some takeaway ideas. I want you to understand that lungs are no longer considered sterile. I want you to understand that most healthy and diseased lung microbiomes consist of these oral commensals, bugs that are found in the oral airway. I want you to know that your lung microbiome is uniquely yours. It is, when you sample your oral airway, it is very similar to recurrent sampling from your oral airway compared to, say, your neighbor's oral microbiome. And I want you to understand that there are two places where we really consider where that influence the lung microbiome, the lung and gut-lung axes that play a role in lung health and disease. And I believe this is the end of my presentation. I'm going to pass the baton on to Dr. Segal to tell you, to have him teach you a little more specific interaction between lung microbiome and NTM.
Okay, so I just want to emphasize one thing, which is that the microbiome includes everything. It includes what we normally is being called commensals, the bugs that live in our cavities, in our bodies, in our mucosas all the time. Includes bacteria that are opportunistic, that may lead to changes, which at times we call it dysbiosis, and includes the pathogens. Some pathogens, some bacteria may be a commensal if they are in the oral cavity, such as Haemophilus, but if they are in the lower airways and they bloom and they grow, they're going to be called a pathogen. So where you are and what those bacteria are doing are going to be determine their role in the pathogenesis. There's no way to eliminate bacteria altogether. There's no way to say, I'm going to get rid of the microbes present in the oral cavity. Indeed, that may not be healthy, actually. And we know that people that in circumstances where you can eliminate bacteria from the gut or from the oral cavity, it actually may do more harm. And the paper that Ben just showed also demonstrate a beneficial role for occasional exposure to microbes in the lower airways, even if they are from microaspiration. Occasionally, it might be okay. The question is, what happens when you have that very, very often in a recurrent way? So in this view, I want to put it in the context of what we know about bronchiectasis. So there are a few things. For a number of years, our focus has been on what we call pathogen. And you can have a bronchiectasis due to Haemophilus, or thought to be associated due to Pseudomonas or Staphylococcus, or non-tuberculous Mycobacterium. But what's interesting is that using that view, using based on culture, which is what I'm talking about, that you identify that your bronchiectasis, in your bronchiectasis, you have this pathogen that grows in a culture media. We know that there are differences in terms of prevalence of these different type of microbes in different places. So this is a display of the prevalence of different type of culture positivity in bronchiectasis in the United States, UK, Belgium, Spain, Hong Kong, and Australia. And you can see that they have different colors. It means that there are different prevalence in different countries. For example, one very significant one is that in the US, NTM is a very frequent and prevalent organism found in bronchiectasis, whereas in the rest of the world it's not that prevalent. Okay. Even if you just look at NTM, even if you just look at non-tuberculous Mycobacterium strains, and you compare the data from two large cohorts, the one in the US that I described at the beginning and the one in Europe, you can see that the prevalence is not the same. It's actually quite different for the different type of strains of non-tuberculous Mycobacterium. So there has to be more. There has to be other things that are contributing to these differences, no? And what I'm showing you here is based on what we know on the culture, based on culture data, no? And this is like, to me, it's like an iceberg, no? This is sea level. You can see the tip of the iceberg, but what you're not seeing is what's underneath the iceberg. That's what you can do when you use culture independence, which is what Dr. Wood described a minute ago. So now we don't rely on culture to grow the bacteria, but actually we rely on studying microbial products that allows us to see the microbes present in a sample in a much more comprehensive and complete way. So what this allows us is to start recognizing many other factors that will affect the microbes present in the lung, okay? And some of these factors are displayed here. So for once, as Dr. Wu said, we have constant exposure to microbes through microaspiration. So this is microbial immigration from the oral cavity, for example, to the lung. So if you have impaired swallows or if you have gastroesophageal reflux, of course, you're going to have more exposure to microbes in the lung. On the other side, there could be some factors in the lung that will determine how much of these microbes are going to stick into the lungs. It could factors such as whether you have a biofilm, whether you have certain pH in the lung, whether you have certain oxygen tension in the lung, and what's the inflammatory status of the lung. And then you have factors that are important to eliminate the microbes, so microbial immigration. So if you have impaired cough, you're not going to be able to get rid of the microbes that goes down. If you have decreased function of cilia, these are some beating hair that the airway epithelium has, that it has to be beating all the time and moving secretions out. And actually, if that doesn't work, and actually there's a genetic condition that affects it, then you have buildup of mucus in the lower airways, and all of that will affect how much the microbes are going to be sticking to the lower airways. If you have problems with your immune system, all of that will determine the microbiome and the relationship with pathogens and immunity. And that is where we think that the investigations needs to go in order to understand why there's such a different type of prevalence of microbes in bronchiectasis and why there are differences in isolation of different non-tuberculous Mycobacterium strains. This is just one example why this is important in bronchiectasis. We know that in patients with bronchiectasis, there is higher prevalence of patients that have GERD, that have gastroesophageal reflux. So that is certainly going to lead to a greater degree of exposure of gastrointestinal and oral secretions into the lower airways. So this cartoon shows different type of bronchiectasis. No, you have your lungs in patients with bronchiectasis can have different degree of structural damage. Some very severe, such as cavitary. Some more affecting different degrees the bronchial tree at a distant level, and some what we call tree and buds, which are, alveolitis, inflammation of the alveoli, and terminal tubes, terminal bronchioles that connect those alveoli. So when you have that situation, you have your microbiome in the lung that all of a sudden becomes dominated by one pathogen. Okay, call it Haemophilus, call it Streptococcus, call it Pseudomonas. You still have your oral cavity microbiome, and there's no way to get rid of it. These are commensals that will live with us. There's no way to limit that. When we take a sample from patients with this, we're hoping that we'll be able to capture that pathogen present in the lower airways that is causing a lot of the inflammatory state. This is an example of this. So we have a patient with bronchiectasis. The culture was Haemophilus influenza. We did this type of, we look at the microbial DNA present in that sample, and we saw that most of it, 70% of it, was coming from Haemophilus influenza. So in that scenario, this is what we think happens. So you have, over time, people get colonized and infected with the respiratory pathogen, Haemophilus, Pseudomonas, and then the amount of bacteria in your lungs goes up, and most of it are going to be that pathogen, Pseudomonas, Haemophilus. So in that condition, you have a pathogen that is causing inflammation. So what are our options? The obvious one that many patients with bronchiectasis is the first thing that they see as treatment is to try to kill the pathogen with antibiotics. And depending on the type of pathogens, we have different selection of antibiotics. An alternative approach that there's growing interest is maybe it's not just sufficient to try to target the pathogen. We also may want to target the inflammatory response caused, because that inflammation can lead to further damage in the lung. There are some now newer therapies. There are some interesting clinical trials that are aiming at doing just that. Now, we have, as Dr. Wood told you, none of these pathogens are in isolation. And we think that there might be events that occur in the microbes present in the lung that precede the development and the acquisition of this pathogen. And that's what we call dysbiosis. And we're very interested in understanding how dysbiosis affect host susceptibility to pathogen. And that's what Dr. Wush has showed you. When we have what we've been showing, what he has just published, shows that an aspiration event leads to increased inflammation in the lung. Okay, and there are different ways in which your lung responds and with the inflammatory cells. In a conceptual model, you recruit different type of cells, such as neutrophils and macrophages. You then, on a second wave, you recruit specialized T cells. These are very smart cells that control inflammation and makes it targeted to different bacteria. But then there's a time to put some brake to stop inflammation, to decrease inflammation, as a way to control it. That immunological brake is also triggered, and of course, after every aspiration event. The implications of this is that, one, this simple aspiration event has implications for the inflammatory tone of your lung. But it's not just controls how much inflammation you have in your lungs, it also has implications for how good your immune system is doing the surveillance against bad bacteria, against pathogen. So if you have too much of this immunological break, this can actually be harmful because you have you're blunting your ability to respond to a pathogen. And that's the line of investigation that I'm pursuing with Dr. Wu. Now, let me go back to this graph of bronchiectasis. Quite commonly, we see this type of bronchiectasis where you have, it's not like a cavitary disease where you have a lot of bacteria in a cavity. You have some more milder, you have more milder images with what we call tree and bud. You have some isolated bronchiectasis. This sometimes is a different phenomenon in which what's happening is that the bacteria, the pathogen, what we call the pathogen, may not be dominating the microbiome in the lung, okay? May, instead of occupying most of what's in the lung microbiome in that area, it occupies a very small percentage. Okay, you still have your oral cavity commensals. When you try to sample and to see what's happening in the lower airways using an induced sputum, this, since it was a small percentage here and the sputum has to go all the way through the oral cavity and when you collect, this might be even a smaller percentage. So the pathogen may represent a very small percentage of what's recovered. Let me give you an example. So this is a patient that I saw many years ago, and we had an infiltrate here. You can see some area for bronchiectasis. You can see a dense infiltrate. We did a bronchoscopy and we obtained a sample from there. The patient, many weeks after being in the hospital, grew up Mycobacterium avium complex. So this is a type of non-tuberculous Mycobacterium. And when we did, this is what was identified by culture. However, if we look at the microbial DNA, we identified that only 0.1% of it was coming from Mycobacterium avium complex. So I'm gonna skip a few slides because I think this is what I want to get to as our understanding progress. In this scenario, what you are having is a pathogen that is not dominant. It doesn't occupy most of the microbiome in the lung, okay? And in that scenario, something different is happening. When we look at what drives inflammation, sure, the pathogen, in this case, non-tuberculous Mycobacterium, may be the driver of inflammation. But all these other microbes present in those lower airway samples can also be contributing to the inflammatory process. And that's what we have revealed in the last few years by using these culture-independent techniques to characterize the lung microbiome. So maybe a novel therapy that we're still not there, but we're exploring is to not just target inflammation, not just target the pathogen, but also targets pathogen, treat the dysbiosis that leads to the increased inflammation and leads to the pathogen acquisition. It is also quite possible that patients have these two scenarios, one where you have a very dominant pathogen, the other one where you have, such as Pseudomonas and Haemophilus, that trend to be very dominant. And And then over time, they go and develop a non-dominant pathogen, such as non-tuberculous mycobacterium. This we see clinically, the patients go back and forth, and why we don't fully understand. So I want to leave you with these two scenarios that are specifically for non-tuberculous mycobacterium. We see a lot of patients with NTM, and many of them have a rather innocuous evolution. They don't have much exacerbations, they don't progress their disease, they don't have much inflammation, and we just watch them, we don't treat them. The other situation is our patients that have the same bacteria, the same non-tuberculous mycobacterium, but they have a lot of inflammation, they have frequent exacerbations. Our investigations are trying to focus whether there are the difference is not just on the pathogen, it's not just on the host inflammatory response, but it's on how this microbiome present in the lung, it differs in those two scenarios. And we think that this can be a very novel and important aspect. So what I'm trying to say is that we're moving away from what's called the Koch postulate. No? The Koch postulate is you have one bacteria that we call it a pathogen, in this case, non-tuberculous mycobacterium, plus some host factors, some factors of people, and the sum of them explains disease. We know that this is not the case for non-tuberculous mycobacterium. Many patients may have non-tuberculous mycobacterium. They don't have any genetic abnormality. Some have disease, some have very, very, very mild disease, okay? Or none, just culture positivity. So we think that we're starting to uncover the role of these other microbes that are present in the lung that may be contributing to why people develop worse disease or not. So why this is important? Because therapy, from the therapeutic level, you can think of many other ways to treat this, not just antibiotics. You can give probiotics that feeds the good bacteria, or that introduces good bacteria, or you can give prebiotics, which is food for bacteria. We're at the very early stages of this, but especially because the lung microbiome is really understudied, but we think that there are important mechanisms that we're dissecting. So the key message is that this lung microbiome in subject with bronchiectasis are going to be is very heterogeneous. Different patients will have very different ones, and we need to dissect how this is playing a role, and I think that the growth in this molecular diagnosis is just changing the field dramatically. Where we're going is we need to stratify patients, considering different aspects, the unique aspect in terms of their clinical presentation with phenotypes, in terms of their unique host immune response, that's what we call endotypes, but also understand microbial dynamics. Not just focus on pathogens, but actually focus on these other microbes that we call the microbiome. What's interesting for me in non-tuberculous mycobacterium is because it's a non-dominant pathogen. So these other microbes are constantly playing a role in the interaction with the host, and we think that plays a major role. Where we're going, where we have a large cohort of patients where we have longitudinal samples, and we have a lot of bronchoscopy samples that are going to be key to dissect some of these factors. I just want to finish by thanking many of the people working in my lab that you can see displayed here, collaborators in NYU and in other academic centers. And finally, I just want to say that the growth of the program and the ability to do this would not have been possible without the support that we got from many donors, which are named here. I also have to say NTM IR has provided us with funding support in the past and has been a wonderful help to advance the research agenda that we're taking on. Um, and we don't just feed our research agenda. We also use this to promote education among patients. We have an NTM patient education program, and that is going to be virtual in October 27. But we also bring the top scientists on this area every year for a scientific symposium, and this year is going to be in December 17. With that, I just want to thank you all for your attention, and I'll be happy to address any questions that the group may have. Thank you.
Thank you very much, Dr. Segal and Dr. Wu. We are going to take some questions now. We have some questions in the queue. First up, somebody is asking, can oral hygiene practices change the lower airway microbiome?
Yeah, no, that's an excellent question. As I said before, it's impossible to get rid of microbes in the oral cavity, okay? You can reduce the amount of bacteria with some practices, but you don't get rid of them. They grow back. And actually, they are important for our oral health. So now the question is, for people that have very, very frequent microaspiration, which we think is bad, maybe a little bit of microaspiration might be okay, but very frequent might be bad. If you were to do something to your oral microbiome, will it affect the lower airways? We just don't know that yet. This is studying the lung microbiome is quite complex because of accessibility issues, but that's certainly something that we're very interested in looking at.
Okay, thank you. So we have somebody asking about a GI test, which is the esophageal manometry and Bravo capsule. If that test proves negative for silent reflux, does the gut-lung relationship, is that still related to GERD or GI? Would there still be a relationship there? Is there something else that should be looked for, perhaps?
Yeah, Ben, you want to respond to that?
Yeah, sure. So that's a great question, and I think it goes to show that, like Dr. Segal said, this whole sort of interaction is very complex, and the gut-lung axis is more along the lines of these communications through small molecules. So there's these things called short-chain fatty acids, and these are byproducts of bacterial metabolism. Just bacteria produce them as a fuel source. They use them, and they can inform, and they can go into your bloodstream and immunomodulate distant sites, including the lung. So I believe this person was asking specifically about whether or not they should worry about GERD or reflux causing changes in their lung microbiome. And I think the overall understanding of direct communication, meaning bacterial bugs coming down into the lungs, we generally accept now that it's mainly the oral cavity is likely the source and not necessarily GERD. That said, there are particular people who may have a lot of reflux from GERD that do aspirate. So, it's a fine line, and I want to hedge what I say very carefully. So it's unfortunately a very complex question, and I hope that answers your question.
Yeah, but I think if you have a negative study for gastroesophageal reflux, at least you have that out of the equation, no?
Yeah.
You can still have aspiration of oral secretions, and that may be playing a role. There are different studies for that, but if you have evaluated your GERD, that's you can feel confident that at least that is taken care of.
No, I agree.
Thank you. We have a couple of questions just quickly for definition purposes, and I think it'll be helpful to some of the attendees. Can you define an aspiration and also a microaspiration and maybe help distinguish the two?
Yeah. Ben, you want to respond to that?
Sure. And in our research, aspiration events, at least clinically, and also clinical and research are kind of two different things. Aspiration events are when things like food and oral secretion, thick oral secretions or upper airway secretions get aspirated, things that large sort of foodstuffs that go into the lungs. Microaspiration is a different phenomenon, we think, and it's a little less defined. We think the fact that we often speak and we breathe, we dislodge these microbes that exist along our teeth and our oral cavity, and they go into our lungs. Bob Dixon at University of Michigan looked into this question in healthy volunteers, where he basically bronchoscopied them and sampled their lower airway microbiome. And where he saw the most similarity to the upper airway are places where micro droplets, very small droplets of saliva or moisture deposit. Places like the carina, where basically where the trachea splits into the two main airway conducting airways. And also, surprisingly, in the alveolus, where a lot of bacteria may be swept there to be cleaned up, perhaps. Yeah. So yeah, an aspiration is much more kind of a larger event, a larger volume event.
Yeah, in other words, microaspiration may happen without not noticing at all. It's such a small, tiny amount of secretions that gets into the lung, and that's probably common in everybody. Everybody has it to a certain extent. Aspiration is a major event where, where people have aspirated a large amount of secretions, and that is obvious and diagnosed easily in a patient that is conscious.
Thank you. Another question, we have a bunch of questions relating to probiotics. So I'm going to start with some of them and see where we can get to from there. One of the questions is, how are probiotics that are administered orally affecting the lung microbiome?
Yeah. And we do not know what, even the probiotic data about how it affects the gut, which is where you're taking it. So you swallow a mixture of microbes which are in these probiotics. Not everybody gets the same effect. Not everybody gets, many of the people ingesting these microbes, they may not, they may kill those microbes very, very rapidly. And we don't understand why. And we think that that's one of the reasons why probiotics in many disease conditions have not been proven to be that effective or successful. Much less is known about what happens with the lung. No, the probiotics don't go into the lung. We don't have probiotics that are delivered to the lower airways. The probiotics are swallowed and go into the GI tract. And the studies have not been done where you evaluate the effects of taking probiotics and look at what happens with the lung microbiome, especially because of difficulty accessing the lower airways and doing this on a patient that takes probiotics. We are interested in our bronchiectasis cohort, where we have over 100 patients, and we are collecting data about whether patients are on probiotics or not. We'll hope to be able to contribute to this, but it's not clear as of yet.
Okay, so let me scroll through some of the questions. We still have some other questions on oral health as well. Somebody has asked a question about antibacterial mouthwash. Is it good or bad for you since it might get rid of good bacteria? They're also asking about saline inhalation.
Yeah, those are excellent questions as well. So the use of chlorhexidine, for example, or mouthwash that reduces the amount of bacteria. We just don't know. We don't know what the effects on lung health are. Basically, because as I say, there's no way to get rid of bacteria. When you use a mouthwash, even when you use hospital grade chlorhexidine, which is a strong antibacterial, you don't get rid of bacteria. You reduce the amount, some bacteria more than others, and actually that, as one of the persons asking a question suggested, that might actually be worse. No, if you don't have control of which bacteria you're getting rid, you might be getting rid of something that is good for you, that is helping. So we're at the infancy of understanding this microbial community use with these culture independent methods. So it's too soon to tell and to give recommendations that people should adopt.
Okay.
Yeah, I kind of think about it like a garden, right? And what you want to do is, if you're using like weed killer or antibiotics that clear out all the bacteria, you don't know what's going to come back. And in some respects, you may get a garden full of weeds following just eradication. Other times, you want to plant a certain kind of bacteria or plant there, but they won't take because the issues with the soil, issues with the nutrients. And it's one of these things we just don't know. It's like Dr. Leo said, it's such a new field. I mean, that there is a lot of potential here, but it's a very, very delicate and complicated thing, manipulating a living thing, and it is this meta-organism that lives in us. And you think, well, what you can do to nurture this garden is just like try to be as healthy as possible, not to have bad habits, and that's part of just taking care of yourself as well.
Thank you. We have a question. What is the genetic consideration that leads to decreased cilia function that Dr. Segal mentioned?
Yeah, so the main one, which is one of the rare diseases to have, is primary cilia dyskinesis, PCD. And it's not very common, but it's one of the, I believe if you come with bronchiectasis, it should be on the list of diseases that people will test you for.
Thank you. Can you better explain the protein content as a seeding condition in bronchiectasis?
Sure. I think, Dr. Segal, this was biofilms that you're referring to. And so there are certain bacteria, once you become colonized, that can form like this film in surfaces, on mucosal surfaces. And what can end up happening is they can change the local environment that this film creates. One of the main bacteria that can do this is Pseudomonas. It's well known to form these biofilms. And there's a hypothesis that once these biofilms form, they actually make it so that it's easier for other bacteria that like this microenvironment to become colonized in this area. Now, this is just some hypothesis, and it's one of the things that we're interested in looking at as well.
Yeah, and there are many mechanisms by which bacteria uses proteins to feed themselves as a source of food. So by different mechanisms, different bacteria take advantage of this. So the more protein content that you have, especially when you have a lot of inflammation, the more food exists for bacteria. So it's like a vicious circle. No, the bacteria induces inflammation. The end product of inflammation is more protein deposition in the airways, the more biofilm you form in the airways, and then the bacteria feeds on it. and helps their growth. So that's a vicious circle that is sometimes quite challenging to cut.
We have a question. Can you comment on CA 19-9, I think is what they said, as a potential biomarker of lung inflammation and NTM treatment effectiveness?
CA 99?
19-9 is what they put. Is that?
The pancreatic cancer biomarker, I think.
Oh, CEA.
CA 19-9.
Yes.
CA is colon cancer, I believe.
Not.
I don't think we're a specialist on this, Leo.
I'm not aware of that being used as a marker.
Okay.
Many of the cancer biomarkers, they go up on the setting of inflammation as well. So, but I cannot speak about this in particular.
Okay. We have several questions on the issue of probiotics again, about brands. Are there any that are currently recommended? Any types?
Yeah, no, there's no probiotic that has been developed for the lung. Probiotics have been developed to be swallowed and go into the GI tract. There's nothing, there has not been any probiotic or prebiotic, which is a different, is giving something that bacteria uses as a food source that has been approved to be used for pulmonary conditions and definitely not for bronchiectasis.
Okay, let's see, there are a bunch of other questions. Let's see. Could you please clarify what treatments you recommend to help with the inflammatory aspect other than antibiotics?
Yeah, so this is been explored for a number of years, and still there are a couple of things. One is actually an antibiotic that has anti-inflammatory properties. That's called azithromycin. Azithromycin, well, it has direct effect to inflammation on top of the antibacterial effect. So it might be that this anti-inflammatory effect might be beneficial for bronchiectasis, and actually it is indicated in patients that have frequent exacerbations with frequent recovery of Haemophilus or Pseudomonas. One of the line of treatment that is available and is accepted is to try to give azithromycin, and it's given every other day on continuous basis with the hope of reducing exacerbations, and there is pretty good data to support it. It is almost standard of care in patients with cystic fibrosis and bronchiectasis, where they take it for many years, especially those that have recovered Pseudomonas. There are now experimental drugs that have come into line in the last year or so that targets neutrophilic inflammation. And there is a paper in the New England Journal of Medicine last year that has shown very promising results. Many of academic centers that I showed you before are part of the consortium that offers that as a part of a clinical trial. So I think it's not that well established yet how to treat inflammation, but I think very interesting things are coming down the line of therapy, and that is worth to pay attention.
I think there was a question here about bacteriophage therapy.
Yes.
That was actually kind of interesting because there are people who are targeting. I think there was a gentleman who was treated with a bacteriophage against Stenotrophomonas. And I think there's a Yale group that's using bacteriophages against Pseudomonas as well. So those are like experimental therapies that are coming down the line that look very promising as well.
Also very promising. The only thing that that you have to keep in mind is that most of the phages are not, even though they are targeted to one bacteria, they may have effects on the other microbes present in the community. And whenever you get rid of a bacteria, there's something else that is going to grow in its place. So that's where we're trying to understand better what are the dynamics between the microbial communities, within the microbial communities, to try to prevent, you get rid of one bad bacteria and maybe you are promoting a different bad bacteria to come in.
Okay. One more question about probiotics. Somebody's asking about potential harm of is there a potential harm that probiotics can cause?
Well, since we don't clearly understand how it works, it's hard to recommend if there's no proven benefit.
Okay.
Now, having said that, most of the probiotics, and there's extensive safety data on probiotics, have not shown to be associated with significant harm. So whenever we have a patient that says, I feel better with this probiotic, I want to take it, we actually encourage if they make them feel better, but we don't have evidence to support it.
Okay. We've had a couple other questions about sinuses. One person is asking if they do a probiotic sinus wash, would that affect their lung microbiome? And then another person is asking about sinus infection and sinus disease. Does that affect the lung microbiome?
Yeah. For some conditions, we know that the nasal cavity and the oral cavity are very different in terms of their microbes present there. And the nasal quite commonly is thought that they can serve as a reservoir for some of the pathogens that eventually leads to infection in the lung. There's data on asthma, there's data in bronchiectasis, where some of the bacteria that is grown into the lungs are very related to some of the bacteria in the nose. Now, a different question is whether there is something to do with the nose, with lavages, that will prevent or will prevent these bacteria from getting into the lower airways. And that, I don't think that we have that great evidence. As I said, any mechanical and most of what we can do in a mucosa, that's not get rid of the microbes completely. It cannot get rid of microbes completely. The nasal cavity, the oral cavity, they're always going to have microbes. So I think it's important to understand how these microbes react and how they interact to better provide a clear recommendation on what to do.
Okay, so we have some questions about dentists, going back to the oral hygiene issues. One person is asking, can tooth infections cause an imbalance in the lung, contributing to increased infection processes in the lungs?
So, say that again, sorry, I missed the question.
If someone has a tooth infection, could it cause an imbalance in the lung and contribute to an increased infection process in the lung?
So, it depends on the situation. Oral hygiene is associated with the microbes that people get into the lung. An example is, sometimes people with bad oral hygiene or a real problem, they can grow a lot of anaerobes. And if they aspirate those secretions, they can get an anaerobic aspiration pneumonia with an abscess. So certainly what's in your mouth will impact what's in the lung. So, poor oral hygiene and having a major periodontal disease, that's certainly should be treated. That's not to say that, in somebody that takes care of the oral health, whether they can do something else to try to prevent having lung complications.
So oral hygiene is important for everybody, including our patients. But do you have any tips on, best practices for NTM and bronchiectasis patients when they do visit the dentist? Are there any particular things they should be doing or should be concerned about?
Not anything in particular related to NTM. NTM is actually the non-tuberculous mycobacterium is not a oral commensal. It doesn't commonly affect or colonizes the oral cavity. Most of the recommendations are related to just good practice for oral hygiene, but not specific for NTM.
Here's a great question. Where would you like your research to lead you in 5 years?
Yeah, I think we are uncovering important mechanisms by which this microbiome affects the susceptibility of getting pathogens. So all the questions that you're asking about use of probiotic or changing oral hygiene, that's what we need to answer. But we need to answer with science. So the hope is that we'll identify good microbiota versus bad microbiota, and we can do something to restore a healthier microbiota that will improve the host immune system and that will improve, will make patients resistant to the acquisition of a pathogen such as non-tuberculous mycobacterium. That's the hope. And I think we have a path that we're leading towards these discoveries.
Okay, we have a couple more questions about dental practices, and I think this is an area of concern, probably because of the concern about water getting into lungs.
Yeah.
So one of them is about a water pick. Could water pick increase NTM in the lungs? And should sonic cleaning or which aerosolizes, I guess, from dentists be avoided?
Yeah, we don't know. For the same happens with being in pools and high mist areas. For some of our more recalcitrant NTM, we recommend that they try, if they do it, to use boiled water to try to decrease the likelihood that non-tuberculous mycobacteria will be present in the water that they use. I think that's, we don't know if that's going to be effective, but that's a low-risk practice that people may want to consider to do.
Okay. So there are questions about inflammation in the lungs and medications. Are there any medications currently in development or medications already existing that are being explored to reduce chronic inflammation in the lungs?
Yeah, so there is this inhibitor of an enzyme that leads to neutrophil recruitment and traps that is being tested. So as that was the paper that I refer from the New England Journal last year. The last author is, or actually the first author is James Chalmers from the UK. And that is phase 2 safety, but showed very promising clinical results. So now we are part of a consortium that is doing a large clinical trials to demonstrate efficiency. Hopefully, if that goes well and it shows positive results, then it may quickly become FDA approved.
Okay, just quickly going back to the sinus question, because there's another question. Somebody's asking, can NTMs be in sinuses? I guess, could they be present in sinuses? Could they cause sinus infection?
Rare, very, very rare.
Okay, thank you. So Let's see, there's a number of, there's questions on other things we've, can you address quickly, here's a question about inflammation again. How is the degree of inflammation in the lungs measured? Can you give
Yeah,
us some insight?
so the way we do it is we do, these are research methods. Now we measure inflammatory cytokines, inflammatory cells. These are not tests that you can do in a regular laboratory. We look at transcripts, which is RNA from the host, from the human cells. But there are no standard ways to do it on clinical, under clinical conditions, also under regular clinical conditions.
Okay. Somebody is asking a question about aspiration and choking. Can you just sort of define the differences between the two?
Yeah, choking is in general considered when you have, a solid piece of something in the airway that blocks the passage of air. Aspiration, and that is a physical blockage that needs to be removed. Aspiration is in general related to fluid or something that doesn't get stuck into the upper airways, but rather goes and travels into the lower airways, carrying, substances and carrying microbes into the lower airways. And those microbes can lead to more inflammation or can lead to a pneumonia.
Somebody else is asking, why do we assume microbes that we aspirate are harmful rather than helpful?
Because it's not black or white. Some microbes, as the paper that Dr. Wu presented, that we just published, in very small amount can lead to some beneficial effects. But if you have a lot of microbes and recurrent aspiration events, that is probably not good. That is probably affecting your immune sensing of an immune capacity to respond to pathogens. And that will lead to actual increased susceptibility to a pathogen.
If MAC is a non-dominant pathogen, would it make sense to treat inflammation that's found in the lungs before starting any kind of standard multi-drug treatment for MAC?
We do not know.
Okay.
And that's why we need to understand better what drives inflammation in that setting.
Okay. Here's another question about microaspiration. Would it be better to try to nose breathe versus mouth breathe to decrease microaspiration, or would that make no difference at all?
I don't think it makes a difference. It's a presumed phenomenon that we do it all the time, and I don't want to promote that nose or mouth breathing is the way to go, because we simply don't know. And if this is a phenomenon that's what we think is pretty ubiquitously happening, and the reality is it's hard to measure and it's hard to define. And I think it's one of these things that we probably do all the time. We just never notice till we found something a marker for it. So I really want your audience not to really prefer one method of breathing over another.
Okay.
Amy, I just have to hop into another call. Ben, can you stay a little bit longer?
Yeah, yes, of course.
I think we have one or two more questions related to the microbiome. Dr. Segal, thank you very much.
Thank you all. I'm happy to respond questions if they send me via.
Thank you. Thank you.
I got big shoes to fill.
So this question is, the microbiome, or specifically the bacteriome, detects both living and dead bacteria. Even if a group of bacteria show high levels by microbiome, they could all be dead. So is the microbiome clinically significant, or should culture still be gold standard, or both are needed?
This is a great question. And actually, when I started working with Dr. Segal, I was also a skeptic of the microbiome. To answer the question, I believe both will be necessary, but currently the gold standard is culture data. And I think that we are starting to understand a lot of these signals that travel with NTM. And to also answer in 5 years where I see our research headed, I want to find some way of treatment for this. I believe that the community of bacteria that travel with NTM is important. We just don't know how to manipulate this right now.
Okay. We have a lot of questions on GERD, so I have a feeling we're going to be doing a whole other session on GERD
GERD
separately.
is very important to this pathogenesis, and I think that'll be great for folks who are interested in it.
Yeah, I think we're going to do that. Here's another question, and I think we'll make this the last one, but this is again about probiotics and the microbiome. A fairly recent study showed oral probiotics delayed microbiome recovery from antibiotic usage by 2 weeks compared to the subjects taking no probiotics. Should we concentrate on feeding our various microbiome using prebiotic foods? Now, I don't know what study this is, and you know, there's no reference to the study, so I have no idea what this is, if it's peer reviewed.
Right, and I greatly appreciate the question, and I'd have to see what exactly this the study was as well. Recovery from changes in your microbiome is very important because we believe in general that usually your microbiome is pretty resilient, and it's built in a way that it probably is supposed to prevent other bacteria, undesirable bacteria, let's say, potential pathogens, from taking root. Sorry for using that metaphor again, in certain surfaces in your mucosa. And I can't speak to specifically the use of prebiotics. In fact, I think there is even less research into prebiotics than there is of probiotics. And I could just say in general that I would want to see more studies on this to understand whether or not the question that the audience member was asking, whether prebiotics should be used instead of probiotics, should be promoted, and what those prebiotics are, what can exactly stimulate the growth or the colonization of healthy microbiota. It's unknown. You know, this is a large black box, and we're starting to get answers, but very slowly, unfortunately.
Thank you. Thank you very much for taking the time to answer so many questions. We really appreciate it. On behalf of NTM Info and Research, thank you all for joining us today. We're delighted that you all took the time to join us. And we look forward to seeing you all next time. And our thanks again to our wonderful guest speakers, Dr. Segal and Dr. Wu. I think we learned an enormous amount of information today, and it was an absolutely fascinating topic. So thank you. And thank you everybody for joining us. Everyone have a wonderful week, and we will see you next time for Science on the Sofa.